Hello, welcome to the Short Read Workshop Day 1 video for assessing library quality. So I've already talked a little bit about library quality control in terms of what issues you might run into. Now I'm going to talk more about how you can assess these issues before sequencing and what software you can use after sequencing. So the first issue that you might run into is contamination, whether it's organismal contamination, nucleic acids such as ribosomal RNA contamination, or adapter dimer contamination. And the most useful tool before sequencing that you can use to determine if you have contamination from these sources is a bioanalyzer or other gel electrophoresis system. After sequencing, you can use a program called FastQC to assess contamination. Base diversity, as I said, is something that you need to be aware of in terms of planning what your library should look like, but it's not something you can assess very well before sequencing. After sequencing, you can look at this also with FastQC. Insert size is something you can assess before sequencing with a bioanalyzer or electrophoresis system, and after sequencing with programs like Picard tools. Complexity of your library is also something that's a little bit hard to assess before sequencing. After sequencing, there's a variety of tools that you can use to look at complexity. And then looking at the concentration of your library before sequencing, you want to use something that's a little bit more accurate than a nanodrop. So a sensitive fluorimeter like a Qubit or qPCR will let you get a pretty good estimate of the concentration of your library. After sequencing, you can actually look at the luminostats and the clustering density that comes out from the sequencing run. I want to talk a little bit about the bioanalyzer, which is one type of automated microfluidic or electrophoresis system. There's other types of electrophoresis systems out there that do something a little similar, but this is one that we've specifically used in our core. And what this does is it's a real-time gel electrophoresis, so you can see signal for the double-stranded DNA that you're getting in your library, and the instrument can convert that signal over time measurements to size estimates. So if you're looking for, say, a general RNA-seq library, this is an output from a bioanalyzer that shows a pretty good generic library. There's a minimum standard and a maximum standard that is run through the machine, and the rest is the actual signal from this library. And what you can see is that most of these inserts are between 200 and less than 1,000 bases, which is exactly what you want for an RNA-seq library. There's also no peaks in any other spot on this report. There's just one major hump that shows a pretty good distribution within the 200 to say 900 bases of this library. Here's a couple traces of libraries that have problems with them. This is an example trace of a library that has major adapter dimer contamination at this peak here. This shows adapter dimers. This library needs to be cleaned up in order to remove the adapter dimers before sequencing. Otherwise, you're going to get a lot of garbage reads and waste a lot of your sequencing depth. Here's a library that shows problems with overamplification or potentially fragmentation or enrichment, in which you have a nice general distribution, but you have a really large trailing hump here that shows that there's a lot of long sequences in your library that really shouldn't be there. And this could be due to overamplification or problems with fragmentation. The bioanalyzer can also easily tell you if you have no library at all, in which case you should not sequence. Now I'm going to mention a Qubit. A Qubit is one type of standardized sensitive fluorimeter. Again, there's other types of instruments out there, but this is one that we've used at BioFrontiers for many years, and it works very well for quantitating library concentration. Another thing that you can do for quantification is qPCR with primers for the P5, P7Ns. Both methods aren't sensitive to enrichment problems in your library, but they are good for quantification, and many facilities will require several different methods for quantification, such as these two. So that's just a little overview of the things you can do before sequencing to assess library quality. We're going to talk more in class about what FastQC and some of these other tools are going to be able to tell you about your library and about the quality of your library. And later in the week, you'll learn how to run FastQC, so you'll become more familiar with these tools as we go on through the class.